As we have discussed in our previous videos, that there are two types of vitamins, water-soluble vitamin and fat-soluble vitamins. Fat-soluble vitamins includes vitamin K, vitamin A, vitamin D, and vitamin E. You can remember fat-soluble vitamins as K, A, D, E. Let's discuss vitamin K in detail. Coagulation is a Danish word, symbolizing its ability to coagulate blood. Moving to the types of vitamin K, vitamin K1, also called phylloquinone, which is obtained from vegetable oils, green leafy vegetables, such as cabbage, spinach, and cauliflower. Vitamin K2, also called menaquinone, synthesized from flora of the large intestine. While vitamin K3, also called menadione, it's the synthetic form of vitamin K. Daily recommended dose of vitamin K in female is 90 micrograms, and in male is 120 micrograms. Let's understand the difference in structures of vitamin K1, vitamin K2, and vitamin K3. This is a structure of vitamin K3, or menadione, with no side chain. Structure of vitamin K2 is same as menadione, but side chain is also present. This is structure of vitamin K1. It also have a side chain. The main difference between the structures of vitamin K1 and vitamin K2 is in their side chains. In vitamin K1, side chain has one double bond, but inside chain of vitamin K2, 4 double bonds are present. Again, consider the structure of menadione. In structure of hydroquinone, 2 OH groups replace 2 oxygens from the structure of menadione, and a side chain is also present, so hydroquinone is formed. In structure of epoxide, 3 oxygen atoms are present, as 2 OH groups are replaced by 2 oxygens from structure of hydroquinone. It also has a side chain, so epoxide is formed. In structure of quinone, 1 oxygen atom is removed from structure of epoxide, so quinone will form. Before moving into biochemical functions of vitamin K, let's discuss the vitamin K epoxide cycle. Vitamin K is essential for activity of vitamin K-dependent gamma-glutamyl carboxylase, which is responsible for post-translational modification of glutamyl residues, or GLU, to gamma-carboxylate glutamyl residues, or GLA, producing a small family of vitamin K-dependent proteins, or VKD proteins. Well, let me make it easy for you through the vitamin K epoxide cycle. Vitamin K-dependent proteins involves factor 2, factor 7, factor 9, and factor 10. You can remember vitamin K-dependent proteins by a mnemonic 2 plus 7 is 9, not 10. Precursor prothrombin, or factor 2, precursor factor 7, precursor factor 9, and precursor factor 10. These are activated and converted to prothrombin, or factor 2, factor 7, factor 9, factor 10. When glutamate residues carboxylate to gamma-carboxyglutamate through an enzyme gamma-glutamyl carboxylase and carbon dioxide, you can see carbon dioxide incorporated in the structure, and oxygen is also involved, which will produce water, but this conversion is incomplete without vitamin K. Vitamin K is cofactor for gamma-glutamyl carboxylase, so which form of vitamin K is used? Well, vitamin KH2, also called hydroquinone, is converted to epoxide. This epoxide can be, again, utilized as this epoxide is converted to quinone through an enzyme called vitamin K epoxide reductase, and quinone is again converted to hydroquinone through the same enzyme called vitamin K epoxide reductase. Let's understand the impact of carboxylation of precursors. So due to this carboxylation, more negativity will develop, and as calcium has positive charge, so due to attraction of negative and positive charge, calcium will attach to prothrombin and prothrombin is converted to thrombin, thus clotting takes place. This blood clotting is one of the biochemical functions of vitamin K. Now, how does warfarin work? There is similarity in basic nucleus of vitamin K and warfarin. So warfarin inhibits enzyme called vitamin K epoxide reductase. Osteocalcin is a calcium-binding protein present in the bone. Vitamin K is also required for carboxylation of glutamic acid residue of osteocalcin. It is involved in regulation of bone mineralization. Moving to the toxicity of vitamin K. So, toxicity with high doses of phylloquinone and menaquinone has not been reported. However, intravenous menadione causes oxidative damage to red cell membranes, that is hemolysis. Diseases caused by vitamin K deficiency are hemorrhagic disease of the newborn. In this disease, placental transfer of vitamin K is inefficient, so deficiency can occur, resulting in neonatal hemorrhage. Newborns have a sterile intestinal tract, and there is therefore no bacterial source. Breast milk is a poor source of vitamin K. Osteoporosis. Recent research has investigated an association between osteoporotic fracture and vitamin K. In diagnostic test for deficiency, we have to measure undercarboxylated GLA proteins in the blood. Let's discuss a case study to have a better understanding. A 72-year-old woman presents to your clinic for follow-up of her ulcerative colitis and diabetes. Since her last visit, she reports that she developed a foot ulcer, which is being treated with broad-spectrum antibiotics. On physical examination, you notice that she has several bruises on her arms and legs, which she attributes to clumsiness. You order routine lab tests, which reveal a prolonged prothrombin time and partial thromboplastin time. You suspect that these abnormal lab values are due to her current medical conditions, and they will likely be corrected rapidly with treatment. Now, what should be the diagnosis of the patient? Patient is suffering from vitamin K deficiency indicated by prolonged prothrombin time and partial thromboplastin time of patient. What are etiological causes of vitamin K deficiency? Vitamin K deficiency is caused by fat malabsorption syndromes and use of broad-spectrum antibiotics, which serve to suppress bowel bacterial flora, thereby decreasing the synthesis of vitamin K, and how the patient with vitamin K deficiency should be treated. Patient should be given vitamin K supplementation. That was all about the vitamin K. Stay tuned to sqadia.com for more such precise and knowledge-stuffed videos.